are looking at the various aspects of this big data and the omics technologies. So which and the challenges starts from your getting your clinical sample, then you're generating your data, then analyzing it, then storing, and then final interpretation it. So as we move on through this, uh, so as we move on through the different stages of identifying our markers for a particular disease. So we go from large scale MSMS proteomics to the finding of the panel of proteins through different feature extraction and the selection. Then the major chunk of the thing is to remain in the validation of those particular proteins, and then finally the data storage and accessibility. But apart from that, so today, which we will be looking, which we had looked into some extent in the previous week. So, but we will today look at the different popular resources that have been built upon to help accelerate and foster the development of the different biomarkers across. So for that, there are different tools that have been developed which help to understand that how we can take it forward. So we will discuss in detail the first of all, like the uh, expression map of the proteins, the maps, the what is your original level or basal level expression in particular tissues, particular cells, particular uh, in the disease based thing, so that one can get an idea that what is actually happening in the disease for better understanding of the nature of the disease and how it can be tackled. So for that, there are different popular resources that have been developed and created so that one can go ahead and look forward for it. So now we will be discussing today mostly the different popular resources that can be used and that is being used heavily for different reliable biomarker identifications. So this is one of the, so as you all, as we are continuously stating the fact that after the human genome project, so there have been continuous evolution in this field of omics to find out the actual level changes from starting from the genomic level to the effector molecules at the particular metabolite level. So here one step which was taken a leap forward for the mass spectrometry based draft of the human proteome. So there was no such, so there was no such thing or how to understand. So because you have all the gene sequences and you have got the genomic sequences and everything, but now you need to find and identify that how the proteins are there and what are the proteins that are present at the human level. So this is how was the first time done using the mass spectrometry base. So as we had discussed that it can be in two different aspects. That is one in your against your one is against your you can try to identify your proteins like in the using the antibody based approaches. Another thing is that to have a mass spectrometry based approaches to find and identify your proteins. So this is one of the group that has tried to do it using the mass spectrometry based draft of the human proteome. So here you can see that it provided you the chromosomal coverage of 18,097 proteins identified in the study. Blue bars your density of proteins in your particular chromosome region. So in the particular chromosome of the particular, for example, this all the 23 chromosomes. So what your how much is your density in that particular chromosome region? And then is the concept of the missing proteome, that is the proteins that are not identified. So we will discuss this in detail as well, that what are the concepts of the missing proteome, that how many types of proteins are there. So so the still there are some proteins for which the evidence we are not clear of their evidence. There are some evidence at the transcript level or at the homology level, but still we are not able to identify or understand those proteins. So this also rise a dark region where one can try to find and explore the missing proteomes. So then using the GPCR proteins as a measure underrepresented in your proteomic experiments. Okay, so this is a they have tried to, this group have tried to identify your, perform the draft map of the human proteome using mass spectrometry. So now, as we are looking, as I mentioned, that we will be mostly looking today in the popular repositories that are present. So once the data is generated, so then we had extensively discussed regarding that how the data can be or should be uh, uh, shared among the shared among the scientific community for better understanding, for better reanalyzing and better interpretation of the data. Similarly, there have been popular public repositories that have come up to make ease of those process and to help or aid in identifying and in doing the things through which you can skip a lot of part, like your literature search or many experiments, through which you can easily use those resources and can come to a conclusion and take your experiments further forward. So proteomics DB is one such kind of tool, so we will be looking into it. So here what is there, so they have taken public raw data repositories, then your in-house experiments. So in-house experiments they have done DNA tissues, body fluids, cell lines, affinity purifications, and then they have uh, kept it in proteomics DB. So this is for the workflow that they have undertaken. So that's why it becomes very crucial. And so not only the uploading your raw data is not only from your publication or your point of view that the reviewers will access or to claim that your data is authentic, but it also gives an opportunity for the researchers across to revisit your data, reanalyze your data, and try to come up with newer explanations or interpretations, as we have already mentioned. So here you can see that the proteome exchange, I believe that you all remember that we had discussed this thing in detail regarding proteome exchange in peptide atlas, massive pride. So all this ecosystem helps us to have those data that are published and that are being generated every day, and then we can further take this forward. So proteomics DB has helped in trying to find out, has incorporated all those data sets. So here you can see that the experimental workflow that how they did for the identification and the quantification of the proteins consists of a repository part of raw data storage designed for the storage, analysis, and visualization of the proteomics data sets. Okay, so this is how they are fully uh, developed it. So now if you go, so this is one of the what you say resources. So proteomics DB is that which helps you. So it started as your protein centric database for the exploration of the large collection of quantity mass spectrometry based proteomics dataset. So from the starting of the week one till today, so we have discussed a large number of resources. So from where you can, if you are curious enough or you are want to analyze on your own, so without having a proper lab setup or proper having proper equipment, you can try on your own, try to search for the data, mine for the data, analyze, and then you can interpret without having much difficulty. So proteomics DB is another resource in that line which will help you to better understanding for your certain question regarding certain protein or assay. So now these data types and contents, then they grew over time to include your RNA-seq expression, drug target interactions, and your cell line viability data. Okay, so this is, and also they are trying to include newer versions. So now let us go and search. We'll see that how proteomics DB is working and what are the different aspects that can be used. Okay, so if it would also be good, so if you all also can go and search it and look for it. Okay, so now this is the interface of their how what are there. So you can upload your own data as well to find out that how analytics and visualization tools on your data using there. So you can have CSV files, so you can have expression data. Okay, so you can, so whatever we had shown in the metabo analyst or in another tools that you can that you do, so here also you can do, but it is still limited with limited access, so mostly you can try to identify and visualize the data. Then you have your peptide level data, then you have your full proteome data upload. So so here you can try to visualize and understand your peptide level data as well. But let us go here. So here, as you can you have seen that in the paper they had mentioned that they are including other organisms as well, but we will be looking for the Homo sapiens only. So if you discover a random protein, okay, so I think you all are aware that you can go and find, if you search in the Uniprot, so you can also get all this information. But here what you can get more information, so here you can see that you can also go directly to Uniprot to find out. So but here if you go and find out the sequence coverage, so in the Uniprot, if you go, you will get all the information like functions, catalytic activity, etc., etc. So now, but on the basis of the Uniprot, on the making it as a base, so now how proteomics DB is evolved. So in the biological, there are two types of databases. So I think I have not touched upon this part. So one is your always your primary database, which contains your raw data like your NCBI, DDBJ, or your PDB, Uniprot. So all this uh, contains your kind of a raw data, raw FASTA files. But then comes your derived databases, which contains from that. So in the biology or bioinformatics, the major problem is the major thing is that only searching a thing doesn't help us, right? So if you just go on and search for a sequence for a particular gene, so it won't be a very much use for us. So for example, now if you uh, so if you are looking at any virus outbreak, so I think that you all are aware now that how much these outbreaks are happening all with vector-borne diseases like dengue or any kind of viral diseases. But so if you just search for the sequence, it won't be much helpful. But you require to perform or identify certain kinds of things in a biological database to understand that whether it is evolving, whether there is a mutation or not, whether the drug that you were giving in the previous outbreak this year it would work or not. So this kind of analysis we need to do. So these all databases comes under your derived databases or the secondary databases, which uh, uses the data from your primary database and then perform some kind of analysis for better interpretation. So the here you can see that it has information from the Uniprot, but it gives us the extra information like the sequence coverage. So another thing in, so it is for this protein it is saying undefined. So now so how many sequences can be can be covered by using trypsin. So now you know that we in the proteomics experiments we majorly follow is the bottom up proteomics, where we try to detect the peptides and not the proteins. But uh, So now we need to understand that the proteolytic enzymes can how much cleave a particular sequence which can be detected in mass spec. So that's why the sequence coverage knowing is much important. Then is the PTMs. So that, for example, if you know the PTMs and if you want to identify the PTMs, so it would be you need a particular kind of enrichment which will help you to enrich those peptides for your mass spectrometry analysis. Otherwise, it would be difficult to detect those. So now, if you have, so now this is another thing is the peptide MSMS. So now, as we had discussed, I think I hope that you all remember, or you can definitely go back and check for your previous week. So we had said that the SRM Atlas is one place where we can find out the peptides or the top hits for deciding on which peptides to take forward for validation after we have found out some proteins or the targets, so that which proteins or which peptides can be taken forward for validation. So here you can see that it has given you the peptides for your mostly for your trypsin peptides, that whether how much chance is that for your peptides to get detected. So here you can see that if you go here, so here they have provided their score, proteomics DB score based on certain parameters. Then this is your log Q value, which you can go and try to filter it out based on things. Then the uniqueness. So again, in this proteomics data sets, we should be aware about the uniqueness, because the proteins that we are identifying should be based on your unique peptide. Then only we can be very much confident about the proteins. Then protease is trypsin. So you can see. So I think you all have identified that these are trypsin by looking at the position which has been cut. So after K and R, there are some lyc as well, so you can see. Then PSMs, so the peptide spectral matches and the experiments, the projects, proteotypicity, and the reference spectra. So if you have certain spectra, so you can see. So these are the spectra that is present. So if you choose this kind of peptides which are having your theoretical mass spectra available, having reported in a more number of your projects or experiments, then you will definitely will be more confident in gaining or understanding of those spectra, that okay, so these spectra are so these spectra are this peptide will be coming in your study, and then you can be more confident of it. Then you have your reference peptides. So you can see. So if you go for the quantification methods for proteotypicity, and if you go for level 3, okay, so they have given you in the basis of your rank order, so number of PSMs, number of occurrences, and those. So if you select your, so there is a slight difference. I think if you have noticed that there is a slight difference, there is two trypsin is given, trypsin and trypsin P. So one trypsin is that which can cut cleave after lysine and arginine only if proline is not present. But another thing is that if proline is present, still trypsin can work. Then is your missed cleavages. So how much missed cleavages you will allow? Ideally, it should be 0, that your trypsin doesn't miss out any place where it is not cut. But generally, we allow up to 2 missed cleavages. So you can see that it you are giving, if you cut with your trypsin and the trypsin P, so then you can have a theoretical coverage of 100% for this particular protein, so that it will be, the whole sequence will be covered and will be cut, and that peptide you can try to inject in the mass spectrometer and detect it. So you will get an overview of that. So but this is again a theoretical estimation. So then you will get, for the general FDR estimate, protein FDR estimates with the target and the decoy network. And this is where it is expressed. So you can see that quantification or calculation method. So this is how median protein expression across. So here you have string, cake. So these are all derived from other database. So this is an classic example of derived database where you can take on help from other sequence of the databases, and then you can definitely try out your work here. Then the inhibitors present against this for a particular protein, so you can definitely get to it. So if you want for any other kind of proteins, that is also possible. So here we will look into it. So EGFR or epidermal growth factor is a one kind of protein that is being generally remains highly enriched in different regions, as well as in the uh, different cancers, brain tumor especially. So here you can see that sequence coverage, which is covered and which is not covered. So you can go one by one and try to explore. So this is your peptide MSMS map with the proteomic DB score. So you can sort it out on also on the basis of it. So having the highest proteomics DB score. And you can try to, so which search engine has identified, that is also important. So also which label, if you want to do, for example, iTrack 8. I hope you all remember the label-based quantification methods, which includes your isobaric tags like iTrack or TMT, or cell-based, in vivo-based labeling like Silac. So here you can see that which are your particular peptides that would require. Now, if you go for level 3, okay, so here you can find it out. So this provides us a good and useful resource. So this is multi-omics. So they are coming, but one thing I forgot to show. So they have also a good. They are evolving is for the drug-based interactions. So you can try to find out for any drug. Okay, so in which proteins it is getting affected, okay, and which cell line, for which duration. So they are trying to give you a whole map and understanding of all those things which have been reported for a particular drug, so that you, when you are trying to choose the drug, you know that what is the effect and what, how it is being affected. So this is how proteomics DB has been derived of, and is trying to help in the process, or to streamline your process of choosing drug targets and finally going ahead. So now So you can try it. As of now, you can try both with your proteins and with the drugs and with the identification of the peptides. Yeah. So any question?
Sir, I have a question that the particular method you showed us, is that applicable in case of drug repurposing?
Yeah. So these are all being. So these all can be taken in the aspects in the lights of your drug repurposing for choosing a particular target, then for choosing a particular drug, and then to try and to identify the targets, refine your drug list, understand those things. So there, in this light, you can always think of. So drug repurposing is one of the applications that you can think of while doing this kind of analysis.
So, but don't you think this has a disadvantage? Because if, as prior to my knowledge, this database only acts on the recorded data that has already been done. It won't identify any novel approaches that is not been done or not been recorded in any particular paper or not been submitted to the particular database.
So what I could understand that you were saying that if you for any particular which is not uh, run not
Like for
on the
example,
thing,
you
so
showed
that's
cisplatin.
why it will mostly be helping. Yeah, so it will be mostly helping in designing your all these resources that we are looking. We will be talking. So there are many resources that we'll be talking after. So all have different applications and different utilities, and it depends on user to user along with their use case that how they want to do it. So for this all one aspect of this databases are also like your SRM Atlas or this or peptide atlas is to guide in identifying newer targets or in the new diseases because of the reports and all those to guide that how you can design your experiment further and proceed for your particular uh, for a particular experiment. Yeah, so hopefully I think there are no more doubts. Yeah. So now, as I was mentioning there, so they have shown that so there have been a series of projects after your that have been developed. For example, for from HUPO or Human Proteome Organism, the organization that Human Proteome Project, Chromosome Human CHPP, for example, then others like the Plasma Proteome Project, etc., etc. So the Human Proteome Project was aimed to discover your high stringency data for all the proteins encoded by the human genome. So this was done to accelerate or foster the search for the missing proteins in the human proteomes. So now we will be looking into the concept that uh, what are the missing proteins that are still left. So you can understand that after all these instrumentations and the technologies advancement of the technologies, still there have been cases for the missing proteins that needs to be unearthed or discovered or deciphered. So the human proteome project that aims to discover your high stringency data for all the proteins. So more accurate till that time it was around 18% of the proteins in the human proteome are the missing proteins do not have high stringency evidence. For example, mass spectrometry means the mass till now those proteins could not be detected by your mass spectrometry. The reasons can be many that they don't that have very much low coverage. For that's why I was telling that these databases, like for example in the proteomics DB, you could check that okay whether the what is the coverage for your particular enzyme proteotypicity, that if you use trypsin or if you trypsin P, can you able to cover the whole protein or not, whether you are using any other enzymes, whether you are trying to get the cover or not. how much coverage you are trying to you can get, or how much peptide you are getting. So all these are there. So all these will help you to understand and identify and design those proteins. So that's why it was pretty much required. So this kind of evidences and this kind of understanding helps us to finally decipher those kinds of proteins. So this was done to find out that which do not have those high stringency evidence. So as I mentioned, that the reasons can be many. So there are certain limitations with the mass spec instrumentation, with the process of digestion, then your desalting, etc., etc. While so much additional informations are there. So here they had tried to made some missing protein pd as a community resource to accelerate the discovery and understanding of those missing proteins. So you can go and search this article in your, I think it was published in Nature Communications. So you can try to find out that the missing protein, how it went forward missing protein to understanding of this missing proteins. Now, what are the missing proteins? So there are, so if you go and divide this into 5 levels, so you can see that the protein evidence level, the protein evidence level can be divided into 5 levels. One is P1, one is P2, one is P3, one is P4, and one is P5. So what is your PE1? So PE1 is one which is your evidence, having evidence at the protein level. That is at the mass spectrometry based level or your antibody based approaches level, you are able to detect it. So this gives you a confidence that their protein have been identified. So you know that at the protein exists, and you have a high confidence. So if you can see in the next plot versions, so we will be looking at next plot as well. So I think I'm hopeful that you remember that we had used next plot for the peptide uniqueness thing. That when we are set up our try to set up any assay or try to identify any protein, so we need to check out that which peptide is unique. So NextProt gives you the peptide uniqueness checker to identify those. So you can see that from 2013 to 2016, the percentage increased from 77.7% to 82.4%. So the evidence at the protein level is increasing with the change in the advancement in your instrumentation technique at the sample processing, sample preparation techniques. So this way, the coverage is increasing and the evidence at the protein levels are also increasing. Second is that the evidence at the transcript level only. So if you are getting your evidence at the transcript level only, so that is your PE2. So this is also getting reduced, but there still are those proteins which are detected or understood at evidence at transcript level only, but is not able to detected at the protein level. Second is the P3 level, which is you inferred from homology, which is 2.8% till 2016, uh, which is now getting reduced, that it was just inferred from your homology. Then PE4 is the predicted, PE5 is the uncertain. So these are the 5 different levels at which the proteins are divided into it based on that where the evidence lies. So if the evidence lies at completely at the protein level via mass spectrometry, then it will be at P1 level. If it is at the transcript level, then it is known as your P2 level. If it is present, if it is inferred from homology, then it is at P3 level. If it is predicted, then it is at P4 level, and if it is uncertain, then it is at P5 level. So which comprises of your total 20,000 proteins. So now, so next plot, as I was mentioning, so look at these pictures. So next plot is gives us to identify those particular proteins. So now you can, so that's why I kept it this thing. So you need to understand that not all the proteins. So not all the data that are published or available in the public repositories are of the same quality. So, so this fact has really been captured in your databases, that the user should be able to view all the data to make a judgment on the reliability of the information you is presented with. So another thing, so since as I was mentioning, that is your one is your primary databases, one is the derived databases. So primary databases contains your raw, like the pride or the proteome exchange. So one user who is generating the data, then uploading the data on the same. But we have to be understood one fact, that all the data that is being submitted or present in the uh, all these tools and the databases are not of the highest quality. So one needs to be very much vigilant in choosing those particular proteins or particular anything, and then relying on those information and then going forward. So that's why it tends to overwhelm the user with too much information. So here uh, we are trying to you to provide all the information regarding the different resources that are present, that you may use it accordingly, so that and try to interpret in a much better way. So, and often makes you simply impractical to evaluate it. It requires that all the users have expertise on all the fields. So all these tools and databases are generally developed on the basis that uh, how to overcome the limitations or the flaws of the existing databases. But please do not take in a linear scale that all those resources have come up one after the other. So this is not in a linear range, but the resources that are available. So in an attempt to overcome this problem, they have used with a on a three-tier policy system. One is your gold, which is showing your highest quality data corresponding to error rates of less than your 1%, or silver, which good quality data but corresponding to error rates of less than 5%. So if we go to the next plot, so one use of next plot in peptide uniqueness checker I have already shown you in previous weeks. So here you can find out that you can go and search for only gold, or include the silver with error bars, with some error bars in the sense some error rates. So you can try to find out some of the proteins. So you can see it's given gold, enzymatic activity, what are there. So chemistry and your pathway. So you will be much more confident when you see that it is written in the gold. So you can see the antibody levels, disulfide bond, mature protein, signal peptides, glycosylation, disulfide. So everything you can check it out here. Further, so here you can see that they have mentioned, including your proteomics DB, peptide atlas, and all ATM databases, and you can have your faster sequence as well. So you can see that where are the reference curated publication databases are there, which has reported about these particular proteins. Yes, so you can go and try to find out. So why we emphasized on this to have particular information in gold or in silver, just to get sure of your data quality that you are taking it forward. Again, in Uniprot, there is option of your annotated, it is reviewed or unreviewed. So if you take any protein which is not unreviewed, then you might get some false positives. So all these things you need to keep in mind while trying to explore this thing. But since we have discussed related to this for the missing proteome and the proteome at the different P levels. So P1, P2, and P3, P4, and P5. So there are some proteins which are in the dark regions. This is one of the things. So these are known as your T dark proteins. If by definition, if we have a PubMed score of less than 5, gene arrive score less than equal to 3, and antibodies less than equal to 50. So this is uh, this is a kind of a workflow that uh, we have followed for under identifying certain proteins, certain UP proteins, which having at your different evidence level. So how? So I'll just share the paper as well if you want to go ahead and try to read it, which we have published from our lab. So here we have reported certain UP1 proteins. So here we have followed a workflow to try to give that the structure and the function of some uh, P1 proteins which do not have reported previously structure and function. So this is the workflow, and that is by definition how to understand that whether they comes under T dark or not. So this is, if you go, this is actually the human proteome structure and the function workflow that has given, and we had used the modified version of it. So here it is given in details, which you can go ahead and read and try to find out. So I'll also discuss. So these UP1 proteins. So this is from the next plot. If we try to get those proteins, then there are tools called your UP Finder and the Pharos, which further helps us in understanding at which P level or which level they are. Are they following in the T dark or not? Then iTezer and AlphaFold. So iTezer was the one which uses is a web-based server and uses your you can predict any protein 3D structure, followed by AlphaFold, which has recently come and uses deep learning methods to predict your 3D protein structures, followed by Cofactor helps you to give the understanding of the molecular biological processes that can be associated with those particular proteins based on your sequence and the structure similarities with other sequences. So now, if you just recall your basic bioinformatics, so once you try to identify and understand a protein sequence or a protein structure, so definitely what you do is that trying to so if you just recall from your the Joe Passman method or the God methods, from that time only the researchers and the scientists have tried to assign that the or try to link with the protein sequence and the protein structure, that the databases that the proteins look similar, the functionalities of the proteins are similar, and which proteins functions leads you to the similar protein structure and the similar protein sequence will have similar protein function. So in this way, you can try to identify the different UP1 proteins, and of course you can go through the paper to learn more about it. So this is the UP Finder tool that we just now discussed. So this you can see that it is a bioinformatic tool for the study of uncharacterized proteins based on gene expression correlation and the PageRank algorithm. Okay, so how they have done that? So they have mentioned. So this quite interesting is to look into the PageRank algorithm. So what is the page rank algorithm? So it was developed firstly by the Google to introduce a new and sophisticated algorithm called PageRank to overcome the problem of the term spam in web queries. Okay, so this was so from there it has been used. So those whoever, if you uh, so the inspiration can comes from all the ways, and then only if it is implemented in biological problems, then only these problems can be solved. So uh, so all this leads us in better understanding of those proteins and identification of those missing proteins, etc. So whatever PageRank algorithm was described to implemented in the UP Finder tool. So this has been used in other research fields, including your molecular for ranking genes, proteins, functions. So this is how the so which algorithm so generally is used is the PageRank algorithm, and the prediction of protein functions using the analysis of your PPINs or the protein-protein interaction networks. So all this network and all those things, it depends upon the ranks or the proteins that are functioning accordingly. So that's remain. So several implementations are available to score the huge graphs, and they measure the relevance proportional to its number of incoming links from high ranked pages. So if you want to go into detail, so that it's quite simply, it's just that how much relevance are coming from the web pages proportionality to its different numbers. So you can uh, get all those numbers. So now, so in from that paper itself, we had used these two proteins had reported that time. So now that was that time it was in the T dark. So now if we go to Pharos, so this from this uh, pipeline, if you go there, okay. So now if you search TM6, so here it is in, you can see, right? So PubMed score, antibody count, PPIs. So now it has gone to TBio. So among, so you need to now choose very much judiciously which tool, which software, which database, which web portal you want to choose for which particular study, for what information, which database can give you the what the high quality data, what the sequences, for understanding this kind of concepts of those missing proteins at which kind of protein levels they are, or the protein-protein interaction. So now with all this knowledge of those different tools and resources, so now you can try to choose out and try to find it accordingly. So you can see that here is the T dark. So T dark requirement. So as we had mentioned, as I mentioned in the slide and in the manuscript, so it required is that less than 5, gene IRF's requirement is less than 3, and the antibodies requirement is less than 50. So, but why it had went to T-bio is that the PubMed score has increased. That means the number of times it has been reported has increased. So that means it is getting detected. And so I also they have given these targets that about which virtually nothing is known. Okay, so there in the T dark is whichever proteins remains. So they do not have known drug or small molecule activities and satisfy two or more of the following criteria. And these targets do not have known drug or small molecules and satisfy two or one of the following criteria. The antibody score and the PubMed score has been increased. Then the T came at least one Kimball compound with an activity cutoff of less than 30 nanomolar. So that target when they have when you know a target. So if you are asked to divide proteins at the different levels, so one way of classification is at your PE level, that is protein evidence at your transcript level, at evidence at the protein level. And another thing is that the T bio came and the T clinical. So one is the so most of the proteins on the basis of this, now you can easily understand that it will be in your T came, that the target has at least one chemical compound with an activity output of less than 30 nanomolars and satisfy the proceeding considers the active ligand is 0. And T clinical has at least one approved drug and satisfy that active drug. So here you can see that it is active ligand for this particular protein is 0, active drug for this particular protein is 0, but so it is satisfying the T bio stage now. So it is not in the T dark but in the T bio stage. So more the proteins will be identified, detected, and evidence and the PubMed scores will get increased and the antibodies etc. will be here. So you can get. So you can view the antibodies as well. Okay, so them 6 antibodies, you can see that the different uh, companies have different antibodies against it, uh, be it polyclonal, monoclonal, etc. So for which aspect or which. So if you are really curious to know and that you have identified certain protein and can try to validate, so you can find out. So this was also coming from your antibody PDS, so which gives all those kinds of antibodies of the different that are available against which and for which things you can use for Western blot, electron, IP or immunohistochemistry or FACS, etc. So similarly, let us try out another protein in pharaohs. So you can see this is in the T dark stage. This is remaining in the T dark stage. Okay, so here if we go, why it is in the T dark? These are your targets which virtually nothing is known. So PubMed score is 2.97. So which publications have been gone? So you can get that which are the publications that has referred to it. Okay. So if you then the gene drift, and the antibody count. Okay, so this is for the T dark region, and then you have T bio. So it is not in other stages. So as we have just as I just discussed, and you can find out the definitions. So this is what the development summary. So what are the different stages for your particular thing that you can try to find out, uh, classify those. So now is the next is the eye hazard. So eye hazard was the one previously being used for modeling. So so if you know the concepts of your homology modeling, one is your so if you just give in the FASTA format any protein, so you need to provide your email, password, ID, and then you can see. So one simple experiment what you can try it out on your uh, own is that one is that you just give a protein, identify any particular protein, one which you are interested, take that sequence, paste here, and run itezer and get the sequence. Similarly, for another protein, go from AlphaFold, take that protein sequence and try to match with the similarity. So this is one thing that can be done easily. So there was a concept of the homology modeling which said that you first need to have a sequence. You will do a perform a blast. So how the homology modeling performs is that you will after you're having a protein sequence, you will blast with perform blast with a particular uh, protein sequence. If the sequence similarity matches and it is much more than the 70 to 80%, then on the basis of the regions or the alpha fold or those regions that are present, you will try to predict the structure of that particular protein. But with the AlphaFold, you will get using the deep learning based models, they provide you your data. So this simple exercise, I think all of you can try it out, perform and try it out to get much more understanding of it. So this is the paper which you can uh, go through it as well. So a prediction of your three-dimensional protein structures. So the community-wide critical assessment of structure prediction experiments, where ITASR, so it was one of the most popular and uh, been used for the different for 3D structure prediction of those particular proteins. So similarly, as I mentioned that in the AlphaFold, so AlphaFold just I will show you certain things. So this I am giving you all those things that I mentioned into finding out at those uh, UP1 proteins. So if you go here, so here it gives you a expected position error. So this one thing that you need to keep in mind while you get the structure, that how much is the confidence level. If you look into it very carefully, it will give that it is very high confident low or very low. So if you have a protein sequence with low regions and then you try to identify certain aspects of it on the basis of that predicted structure, then you might be wrong, and then it would be very much difficult for you to take it forward. So that's why I thought of showing you and just making you aware of this particular course or particular things. Okay, so this will give you uh, a better understanding of those particular proteins. So after, so if in the pipeline you go and find out that, so we will going to the sequences, then prediction of the sequence structure, since very less evidence is known, so we have to predict and get the structure. Obviously those structures are not present in PDB, and then finally we are going for the cofactor or finding of the gene ontology or those uh, things. So if you go for your cofactor, so iter or cofactor generally takes time and will provide you the result within a day or so. But definitely that experiment what I told is worth trying also. Here you will, so you can see that human UP1 protein annotation forum. Okay, so here they are given. So prediction for 16 on human chromosome 17. So this was a kind of a project and where they have used and all those things function prediction for 66 proteins. So you can find it out that on the basis of all those. So this is how it generally provides you the different components. So here, so another uh, experiment or another analysis that you can perform, as I mentioned, that one is that you have your protein sequence and trying to find out the structure similarity from itazar and AlphaFold. Second is that the structure functional similarity in cofactor. So you can upload both the PDB files from one from itazar and one from AlphaFold. So that will give you that identification of those that how much the regions or the gene ontology or those molecular processes or the biological processes that you were getting, that how much is the similar or how much is the different. So that's two exercises you can easily do. So so you can understand that how easily with the help of these web-based tools and those tools you can try to explore the different aspects of it. So for example, if you do an analysis from your, so we have already shown you the gene expression omnibus databases, and there you have got certain genes, and also using the different proteomics datasets. If you analyze your data and then try to find out certain proteins, and then try to find out the structural aspects of the, as someone was mentioning about your drug repurposing or the structure-based drug designs and all, to identify the protein structure. And if the protein structure is not available in your uh, PDB structure is not available, you can try out three things. I would say one is your homology modeling on using modeler or anything, or there is a tool Swiss model, or you can then try to find out your uh, using AlphaFold or using itazar, and then try to find out those protein structures and then their functions and all, and also you can try out different protein interactions, etc. So now this I showed you just that how the function prediction for the 66 UP1 protein. So you can just try to find out that if you get some UP1 proteins and using that pipeline which I showed, that you can try to find out or not certain aspects of it. So this is how it is showing. So you can see that all those databases mostly tries to interlink all those databases, so that the derived database or the resource database gets much more comprehensive and it becomes much more helpful for the different researchers to get help of it. So this was one of the things from firstly from the mass spectrometry point of view regarding that. So getting the making the draft map of the human proteome for mass spectrometry, where it depends on various factors. So that's why you need to identify that which peptides and then their proteotricity or those things. But another thing is that the so antibody based evidence or finding of the antibody based proteome of the human being. So this is one thing which project which was launched in a multi-dimensional scale of your human protein atlas, which gives you the a special map of the human proteome on the project from KTH Sweden. So how they have done it? So this human protein atlas is a kind of a comprehensive map with the expression level at the spatial region level of the tissue at the cells. So they have different projects which we would look. Then it in the how much is the RNA-seq level expression level, how much is at the protein expression level. So all those things are uh, present accordingly. So you have the organs, the tissues, then the cells, then the organelles. So you will have RNA-seq data. So this gives you an understanding of the correlation between your RNA-seq and at the protein level directly, that how much sequence, how much expression level is further taking at the proteome level. Then using the immunohistochemistry for tissue and the pathology based atlas, and for the immunofluorescence, the cell atlas, it you can identify. Means they have identified. So this is gives you the human protein atlas thing. So which we will be exploring. So firstly, if we look at how overview of the different things are there in the human protein atlas. First is that for combining different kinds of the proteins, meeting which links to primary data to the sub atlases. So let us go and look into the human protein atlas. So as I mentioned, that the human protein atlas or this kind of comprehensive information helps you to guide you to understand that how what is the basal level expression of those proteins, how the protein comes. Then you finally try to find out that in the disease state how much it is different, and then how it is taking shaping up, and for the disease severity and the disease going becoming aggressiveness or the druggable part, or whether it is actually the drugs are acting or not, or all those things help you to identify. So here you can see that it is. So if we go to the tissue section or the tissue based map of the human proteome, so there this the groups, different groups try to map the human proteome on the using your different technologies. One was using your mass spectrometry based technique, another using your uh, antibody conventional antibody based techniques, which have their own pros and cons with your, if it you I think that you will understand and appreciate that one becoming high throughput, another thing becoming it's cost, so you have to raise antibodies and all those, but the sensitivity and those things are much higher for your antibody based things. So all these things are very much dependent, and both the technologies have uh, their own pros and cons. So this you can see that the expression profiles in the human tissues of your genes, both on the mRNA and the protein level. So 44 normal human tissue type is derived from your antibody based protein profiling using conventional and multiplex immunohistochemistry. So now to understand that if you want to understand at the presence of a particular protein that you have an antibody or not, whether it has been explored or not, whether in the tissue, at the tissue level, what is your expression level. If you want to know all these kinds of facts or all these kinds of analysis, then what you will definitely do is that you will definitely try to go and find out in the human protein atlas. Whereas if you want to know and you want to do a mass spec based experiment and you want to identify the proteotypicity of their enzymes and all. So then you will go to try to find out in the SRM atlas or the peptide atlas or the proteomics DB to for further understanding of those data sets. So for this reason, you need to understand. So final after that, if you identify certain UP1 proteins that you need to characterize, then you will go and find out in the go through that pipeline of the Faros and UP finder and the next prot, and then finally going to the cofactor or the prediction of those things. That if that can be, for example, in a particular disease, you are trying to find out, you are trying and you have find out certain protein that is coming as a very good against those diseases, but that has not been known much for to a great extent. So then definitely you will go and try to find out that whether you can at least predict some information or not about this particular protein. Finally, you can always there is a scope of validating all those things. So all underlying normal tissues with the knowledge based analysis, protein data covers 15,303 genes, of which are available antibodies. So around 15,303 genes, of which there are available uh, antibodies are there, and the mRNA expression from the deep sequencing of RNA set from 40 different normal tissue types. So how protein is localized in tissues at a single cell level. So you can just go and explore that how, if you want, if you are very much interested in a particular thing, how the proteins are exposed, if a gene is enriched in a particular tissue for a specific tissue or not, which genes have a similar across tissues, that is your housekeeping genes. So all these things you are getting. So you can see that eye, heart, muscle, glands, lungs, everything are covered. The tissue enriched, group enriched, pro tissue specificity, and all distribution. And the enriched genes in kidney, in retina, in tongue. Then you have the subproteomes, tissue specific proteome, housekeeping proteome, as I was mentioning that which proteins are there. Then the regulatory proteome, secretin and membrane proteome, isoform proteome, cancer proteome, druggable proteome. So if you just go one by one and explore, human tissue specific proteome, the brain tissue enriched. So you can see that what 75 genes are found which are enriched in the particular tissues, enhanced, pathology enhanced. Okay, so now we'll look into one by one. So housekeeping proteome. So one second. So detected in all the tissues, TPM greater than equal to 1. So I'm hoping that you all remember the concepts of FPKM, RPKM, and TPM. So whenever you will deal with the transcriptomics data sequence and try to find out that what are the expression level, quantifying expression level. So you will know the sequence, the unit like the transcripts per million or the fragments per kilobase million of reads or reads per kilobase million of reads. So if you just uh, don't recall, just go back to week 2, I guess, and you can easily come to the concepts. So RNA polymerases, ribosomal proteins. So all these things are coming under your housekeeping proteome. So if you have a regulatory proteome, so regulation of your gene expression, examples of zinc finger, mucin zipper. So all these things you all know, but you can go and find out their expression levels. Then if you are interested in the cancer proteome, so the number of genes, somatic mutations, and all, and the protein expression. TP53 in colon cancer. In colon, what is its expression level? In colorectal cancer, what is its expression level? So this is uh, one best way to for you to understand the expression levels and can visualize. A major thing is that if the immunofluorescence and the immunostaining concepts, antibody based approaches, is that you can visualize the thing and can appreciate the fact that how it is working. So for example, TP53 in lung. So I'll suggest you all or recommend you all to explore it thoroughly. So this will give you an extreme overview and will aware of you, make aware of you the things that are happening in the lung cancer. So this is ERBB2 in breast and the normal breast and the breast cancers. This EGFR. So as I was mentioning, in the is EGFR in the cerebral cortex and in the glioma, RAS, June. So all these things and your druggable protein. So if you want to define, so a drug exerts its effect by interfering with any four type of macromolecules in the body. So I know that you many of you might be interested in these drugs and all those things, and is also very much of interest in general. So if you have these proteins, polysaccharides, lipids, and nucleic acids, so these are the only four macromolecules on which your drug will uh, work. But almost, so you can see, so almost all your approved drug targets on the market today are direct against protein targets, since issues like toxicity and low specificity are more related to the three letter types, because the lipids and all polysaccharides are present on your cell organelles and on each and every cell. So targeting to particular species or particular cell or particular against any cancer cell, so it becomes naturally becomes quite difficult if you trying to use that, unless you are trying to find out against some marker. So they have already written that there are approximately 20,000 human protein coding genes, but not all your proteins are suitable for drug interactions, and even fewer are appropriate for drug targets. So fraction of the ability to bind a small molecule with required affinity. So as you all might know it, adequate chemical properties, and at the same time potential targets. So this from here they have given the drug targets was like the thyroid stimulating hormone receptor. Okay, used for detection of recurrent thyroid cancer. So how that looks? Gastric LIPF target for uh, some diseases. So how that expression looks. So all these things they have done it very. They are keeping on doing it very comprehensively. So if you just so apart from this, there is your brain single cell type, tissue cell type, pathology type, disease type, interactions, immune cell. So if we go for the tissues only, once again, so if we go for the heart, okay, so you can visualize it in much bigger way as well. Okay, so you will get the from the company, so that you were looking at in the antibody pedia, and then the thing, so you can see, and then the Atlas antibody center group, so you can find it out accordingly. So now we will be looking into the pathology atlas. Okay, so you can see that wherever you are getting the particular expression level, you are it is given pathology enhanced, tissue enhanced, subcellular supported. So the full green means that it is enhanced, which are not available, not available. So you can see that the cytoplasmic expression in the neuronal cells, localized subcellular localization, that it which part of the cells it gets further localized. Predicted location is that intracellular within the cells. So brain neuronal signaling, low human brain regional specificity, neurons misfunctions mainly, cell type enhanced is in this. Prognostic summary, prognostic marker in prognostic cancer, prostate cancer, favorable, low cancer specificity. Another thing is that in the blood, can you able to detect or not? Okay, so also you have your certain downloadable data which you can download. This is the RNA expression, RNA summary. So this will give you that uh, what at what level. So in the human protein atlas, one thing is that if you trying to find out any target on a particular for drug or for anything, so you can go and try to find out that what is the specificity level and what is the uh, expression level across the tissues. So the way where you are targeting the particular protein or the particular gene should be expressed at that region only, so that your drug or those targets can go and bind to those places, and it doesn't affect you, or the cytotoxicity becomes less accordingly. So here you can go and find it out the RNA data for those particular, and then the antibody data. So as we showed you previously. So if you explore it thoroughly, you can get a much comprehensive view of with the cells and the antibodies and everything. So I just kept a brief basic one two slides of that, how those immunohistochemistry is performed, where you have your fixed cell or tissue, and then you have the primary antibody, then you will have the secondary antibody, and you will have the substrate, and then try to find out. So how the tissue sample is done. So after you have collected the tissue samples, so you will do the tissue processing, and then you will in the tissue section, and then you will see the results. So that's what we were seeing the results. So you can visualize it after performing those aspects. So this I just tried to bring in this fact. The thing is that, so the project that was performed for the human protein atlas or the tissue atlas, so it has also been very much contributed by uh, Indian your clinicians, so who have helped in uh, uh, this you're in the immunohistochemistry process, that in visualizing and analyzing your data, and in those sectioning and the staining. So this was very much uh, helped in finding this kind of atlas, which is helping the researchers for better understanding of it. So here you can see that the tissue microarrays and the Indian surgical pathology contribution to the Human Protein Atlas project, which was very much now you can see that how much it has been well documented, and gives you an exact overview of how we want to uh, look into it. So you can see that you will get the RNA expression and the protein expression simultaneously. So both correlation you can get to it. So you can see that there are certain, like the RNA expression is high but no protein expression. Similarly, protein expression is high with very less RNA expression. So there is both positive correlation and the negative correlation, but depending upon how the regulation happens. So that's why now again this comes that you need to understand all the levels in a together way, so that you can finally try to conclude uh, any butter any other things. So this is how you can see that the expression, antibody staining, and the RNA-seq data. Okay, RNA expression overview for that particular thing. In the human cells, so that is during the immunofluorescence. So another thing is the human plasma proteome project of the HUPO that was undertaken. So why human plasma proteome project is requires is that because it holds the potential. So you can understand that having the tissues and having those any invasive kind of things becomes very much difficult. But definitely, if you have the human plasma, having the biomarker from plasma or serum is quite helpful, and in that regard, and it helps you to uh, uh, help in clinical translation in a much better way. So that's why having a human plasma proteome project is quite necessary for in that aspect. So it plasma and all those things, so which are being now also being used. So you can go and read for it for different uh, diseases. So now coming on, so I like to share with you this another resource, so which has been is being actually being actively developed by our lab, and it's being developing. So this is the brain prot, which where so this is from uh, here in IT Bombay, which is being help you to visualize your the different data sets related to the brain diseases. So I'll just give you a demo. So here also you can go and so here you can see that there are all total six domains for related to brain diseases and uh, brain markers. So I'll just give you one domain each, a quick understanding, so that you can go and further explore on it on your own. So you can see that, so this gives you the information about all your particular IDs that are there for your particular disease. For example, Alzheimer's disease, you all know that it is quite popular and quite dangerous disease, or the neurodegenerative disease that occurs mostly in adults and helps you to means elderly people, and this leads to the gradual decline of memory or the cognitive function and your behavioral patterns. So now, so if you are interested to learn about this particular disease, or want to explore more, or for any kind of this kind of disease, which around 50 diseases which are present in the brain plot as of now. So you can go and search your disease and all those IDs which are interlinked diseases. But now, if you want to search that which are the markers or which are the things, yeah, I'll take the question. Just I will finish and then I'll take any question. So first domain is the, if you go, is the BDMC. So yeah, so what you can find out here is that, so here are the different popular, so there are different uh, algorithms, different knowledge bases, different databases having uh, so there are different databases and there are different knowledge bases which can help you to identify your top markers. But here one can visualize using those different algorithms that are giving you the particular score for the markers for the diseases. You can come and try to find out for the particular diseases. So here you can choose accordingly and you can find out. So for example, this is when you are choosing in x-axis, that is generate one of the uh, database or algorithm that gives you the score, and with a y score of harmonism score, you are getting FOE is coming uh, as a top marker. So similarly, you can change and then you can find out that, for example, gene APP and is coming as a uh, top markers. So these are the top 10 markers, which is based on the combined score. So easily at a glance, you are able to identify these particular genes and these particular mm, markers from the based on the basis of your literature data mining. So here you will get the scores along with the BDMC score. So if you just sort it out on the basis of the BDMC score, so you can find out easily find out the markers which are very much coming as relevant for your particular disease, which you can easily go ahead and take it forward. So now, if you are interested, so okay, you have got those markers, but how it actually looks in your real data set. So what you can do is that you will go to the BGTM, and here, for example, APP was coming very much high, uh, significant marker in your particular Alzheimer's disease. So you will go and search. So in the Alzheimer's disease, if you go and see that, so now you will again will appreciate the one. So see the significance level. So again, you will appreciate now that the markers that have been reported. So always getting in this kind of omics data set, the significance or those things is not at all easy, because a lot of variability is induced, because first of all you need to have a good number of in statistical power to have a good under getting the actual biomarkers. Then the during the experiment it becomes very much difficult for you to have different variabilities gets induced, and that's why all those things doesn't come up, but how in the real data looks like, you can get a glimpse of it uh, from here. Then that is your transcriptome, or the microarray based RNA sequence data from where you are getting the BDTM. So now in the BDPM, you can go and search for EGFR as we had looked. So from the public repositories, that how in the publicly available data set, so you can easily go and search, and then you can interpret. So you can see that the EGFR is coming highly upregulated in your disease in glioma or glioblastomas. So this, see, now the problem, the omics data sets, as we were talking and discussing, from like the problem of the sample number, heterogeneity. So all these you can now see and visualize on your own in context of those brain diseases. If you go and brain protein can understand, and also you can try to have more of your data sets. So all these public data sets are helping us to visualize, and this kind of domain helps us to, so helps any researchers or neuroscientists or anyone, for that matter clinicians, so who are not able to analyze and visualize, because that's first of all a time is required, then you need a specific skill set, then you need a specific computational power and everything. So you can, in this kind of resources, helps you to uh, see, visualize, interpret, analyze your data in a very high in a very quick manner, and also helps you to come to a conclusion and design your further experimental studies accordingly. Then is the another last domain is for the uh, BDDF, for the brain disease drug finder. So as I showed you, the druggable proteome or the druggable region. So what are the different drugs that are being going on for a particular disease? So in the clinical trials region, so if you want to find out that which are the drugs, so that you don't reinvent the wheel again, and you can have a knowledge that what is going on actually, and how many are in the which trial phase and completed or not, you can get an idea. Also another idea that so I think you all have noticed in the TKM and the TClean, that if the proteins are in the TKM, at least one chemical compound will be there. So the compounds or the top markers that we are getting, that how it is coming, or what are the different compounds that are reported for it to, so that the drugs are the small molecules that are reported, that you can take forward on the basis of the markers, and that can be of multiple uses. One might be that you are trying to find out your uh, drug, or you can try to find out that by blocking that particular target by using a small molecule inhibitor, that how that is functioning, uh, whether it is giving some uh, effect or not. So you can use it in a cell culture or in vitro based level methods, or you can have it in animal model studies. So here you can see that the, for example, if you, yeah, so you can see that for this particular gene name or the compound, what the top markers that have came. So you can get the Campbell ID, the drug name, the score, and the clinical phase, the toxicity, that whether they are have whether that against that particular uh, molecule, the any toxicity is reported or not, what is the clinical phase. So you can get all those information before starting your designing or experiment of those. So all these tools are helpful in a way that gives you an opportunity to understand the data much well. And also, sorry, one thing, just missed it out, that BrainProt also have this portal of IBPM. So this is the portal. So as I showed you the uh, in the in your uh, human protein atlas, that the basal level protein expression. So this particular domain contains about the uh, basal level expression in a difference in the hemisphere wise and their phosphorylation status, that what is how they are. So you can see that the both that in the basal level in the hemisphere wise what is the difference, as well as in the phosphorylation level what is the difference. So this is which is contained within your brain plot, and it is only resource available for regarding your the difference in the expression level in your hemisphere wise. So from the, so you have the reference from your basal level protein level expression, you have the reference at the literature based evidence, you have the reference at the for any particular disease at the transcriptome map and at the proteome level, and also the drugs or the inhibitors that are present on it. So in a one shot you can try to find out all those things. So yeah, so this led us to the conclusion for this particular week is that a lot remains still now to explore in multi-omics system to understand the landscape. So you can understand there are so many proteins in the T-dark stage till now, which you can try to find out. So these resources are helping us to comprehend the different disease pathophysiology. So we need to explore for better understanding of those deadly diseases. Yeah, so thank you for this. So this was all for this